This video will observe the scientific differences in Jello when using different types of pineapple. The purpose of this lab is to observe whether canned or fresh pineapple is best to use when making pineapple jelly. The materials needed for this lab are 2 400 ml beakers, a 100 ml graduated cylinder, a stirring rod or a spoon, a Petri dish, and a weighing scale. The ingredients needed are fresh pineapple, canned pineapple, Jello powder, hot water from a kettle, cold water, and plastic wrap. The independent variable is the type of pineapple, whether that's fresh or canned. The dependent variable is how well the jelly solidifies, and the controlled variables include the time left for the Jello to set, the temperature of the water, and the type of Jello powder used. If both fresh and canned pineapple's abilities to create pineapple jelly were tested, then the canned pineapple will make a more solid one due to its lack of the enzyme bromelain. The presence of the enzyme will catalyze the breakdown of collagen, which is what the Jello is made up of, preventing the Jello to set. The canned pineapple goes through a heating process, so the bonds will remain intact as they are supposed to, creating a jelly-like structure. First, make sure that both of the beakers are labeled with fresh and canned. In a Petri dish, measure 42 grams of Jello powder and place it into any one of the beakers. It is observed that when pouring the Jello powder, some particles flew out of the Petri dish. This resulted in a loss of the reactant in the solution. In the graduated cylinder, measure out 125 milliliters of hot water from the kettle. For accuracy, there were 2 graduated cylinders used, one with exactly 100 ml of water and one with 25. Pour the measured water into the beaker containing the Jello powder and mix using a stirring rod until all the powder has dissolved. It can be seen that the powder completely dissolved into the water, creating a red homogeneous mixture with a little bit of a fruity smell. Next, again using the graduated cylinder, measure out 125 milliliters of cold water from the tap. Pour the cold water into the Jello mixture from before and stir well. Now measure 125 milliliters of the Jello solution using the graduated cylinder and pour it into the other labeled beaker. There should now be 2 beakers with the same amount of Jello solution. Now, using the electronic balance, measure exactly 51 grams of canned pineapple into the Petri dish. Place the measured pineapple into the beaker labeled canned and using the stirring rod, gently stir the Jello and pineapple mixture. Next, measure 51 grams of the fresh pineapple and place the pineapple into the appropriate beaker. Before stirring, make sure to wash the stirring rod well to avoid cross-contamination. It can be observed that the solid fresh pineapple pieces mostly float to the top of the solution while the crushed pineapple is equally immersed throughout the solution. The fresh pineapple is also much more visible and evident, while the crushed pineapple just makes the entire solution look chunky as a whole. Lastly, cover both the beakers with plastic wrap and place in the refrigerator for 24 hours. Make sure to wash all the materials used and to place them back in the appropriate spot. Water droplets can be seen on the plastic wrap on both of the beakers. The canned pineapple successfully created the correct semi-solid Jello structure. It is shiny and gel-like to the eye and feels very smooth and jiggly to the touch. As shown, the Jello can be broken down easily and has an extremely jiggly structure. The pineapple bits can be seen inside, which makes this a heterogeneous mixture. It was able to hold its shape and had solidified to a jiggle yet condensed state. When flipped upside down, the Jello stayed in the beaker. On the other hand, the fresh pineapple mixture displayed opposite results. It had retained no visible structure other than the slight thickening of the very liquid mixture. When shaken, the contents of the beaker splashed around in a very similar consistency to water but a little bit thicker, and the pineapple chunks were still floating at the top of the solution. Jelly contains gelatin, which is a protein that comes from collagen. Collagen is made up of 3 intertwined amino acids or polypeptides: glycine, proline, and hydroxyproline. These amino acids are intertwined in a secondary structure of a polypeptide, a triple helix with a coiled shape at room temperature, as shown on the screen. When collagen is added to hot water, which is what is done to make Jello, the bonds between the helixes break and it unravels, which causes the mixture to be a liquid solution. When this solution is cooled, the collagen protein chains reconnect, but in a different way than before. This is because cooling proteins causes them to move slower from the decrease in kinetic energy, changing their shape. While the chains are reforming, the water molecules get trapped inside the chains, which is why it turns into a semi-solid product, Jello. Pineapple contains a protease enzyme called bromelain. Enzymes are biological catalysts and they speed up the rate of reactions. Leaving enzymes in foods may cause unwanted enzyme reactions to occur, which is why canned foods are always heated. Heat is a factor that causes enzymes to denature. Every enzyme has an optimal temperature, and when it passes this temperature, it denatures and can no longer be used in reactions. When tinned pineapples are cooked beforehand, the bromelain reaches its optimal temperature, which is 60 degrees, and then denatures, meaning its bonds break, which is why it is no longer present to hydrolyze collagen. Therefore, the Jello mixture is able to solidify and set since there are no enzymes preventing it from doing so. Now, since bromelain is present in the fresh pineapple, it catalyzes the hydrolysis or breakdown of the collagen in the Jello solution while it is cooling in the fridge. So how does it do this and what happens? Enzymes catalyze reactions through the enzyme-substrate complex. In this case, collagen is the substrate while bromelain is the enzyme that breaks collagen down. Since the enzymes in the fresh pineapple were not denatured through a heating process, bromelain was able to bind into collagen's active site and attain an induced fit model. An induced fit model activates the enzyme so that it can perform its catalytic function. The presence of an enzyme means that the activation energy required to complete a reaction decreases, which means that the bromelain causes the breakdown of Jello to occur faster. This hydrolysis caused by bromelain in pineapples is what prevents the Jello from solidifying. When collagen is broken down from the induced fit model, the trapped water molecules are able to escape the chains, which prevents the creation of semi-solid Jello. This is also why it was important to pour and stir the canned pineapple first in the experiment to ensure that no bromelain from the fresh ends up in the canned solution. One source of experimental error in this experiment could be that the temperature of the cold and hot water was not controlled and known exactly. Since the hot water was taken from the kettle without a specific temperature setting on it, the water could have been any temperature, maybe not even boiled enough. This could have affected the experiment because not using boiling water in the Jello solution would have slowed down the altering of the proteins that trap the water molecules. If the exact temperature were known, then more accurate results and observations could have been made. For example, if the water was not hot enough, then that would have resulted in a more jiggly and runny Jello mixture, while if the water was too hot, it would have resulted in a more firm Jello. This is because the hotter the water, the faster the collagen triple helix will unwind, and the faster the water molecules will get trapped inside from the increase in kinetic energy. This will give the water molecules more time to set inside of the collagen, which will result in a more firm Jello. It could have also been observed whether or not the different water temperatures affect bromelain's hydrolysis of collagen, and if these different water temperatures affect the final liquid solution. Therefore, although this experimental error may have caused only slight differences in the results, knowing the exact temperature of the hot and cold water may have slightly affected the final Jello formation. Overall, the hypothesis was proven correct because canned pineapple was able to make a more solid formation of Jello due to its lack of the enzyme bromelain. The presence of bromelain in the fresh pineapple broke down the protein collagen, which is what prevented the formation of proper jelly and resulted in a liquid solution. Therefore, it can be concluded that canned pineapple is the best to use when making pineapple jelly.